Welcome back, everyone, for another deep dive. This time
Mm.
we're looking at, aging research and specifically telomeres.
Ah, yes.
And so this is a really cool article that was published just earlier this year in Nature Communications.
Very cool stuff.
Um, and basically, researchers were able to create this new strain of mice, and they're calling them hut mice. And essentially, they've given these mice telomeres that are regulated just like yours and mine.
Exactly. So the telomeres in these mice end up being a similar length to human telomeres.
Yeah, so that's the big question. Like, how did they pull this off?
Mm.
You know, mice and humans are different in a lot of ways,
Yeah, for
and
sure.
telomeres are definitely one of them.
Right.
So maybe just as a quick refresher for folks, can you kind of remind us what telomeres are and why they're so important for aging?
Yeah, so telomeres are like those little caps on the ends of our chromosomes. They're like the plastic tips on your shoelaces that keep them from fraying. Every time a cell divides, those telomeres get a little bit shorter. And eventually they get so short that the cell just can't divide anymore, and that contributes to aging.
Gotcha. So it's like a biological clock ticking away in every cell.
Exactly.
Okay, but back to the mice. You know, I know that mice have much longer telomeres than humans do.
Right.
So how do researchers actually study aging in humans using mice when they're so different?
That's one of the limitations, right? Because mice just don't age the same way we do.
Right.
Their telomeres are much longer for one. And they also have this enzyme called telomerase.
Oh, yeah, telomerase, that's the one that can actually rebuild the telomere.
Exactly. And it's active in a lot more tissues in mice compared to humans.
So using mice to study human aging, particularly when it comes to telomeres, isn't always the most accurate approach.
Yeah, it's like comparing apples and oranges in some ways. So that's why this new hut mouse model is so exciting.
So how did they actually create these hut mice? Did they, like, give the mice genes for shorter telomeres?
Not quite. They did something even more clever. They took the mouse tert gene, the one that makes telomerase, and they swapped out some of the non-coding regions with human versions of those regions.
Okay, hold on. Non-coding regions. For people who aren't geneticists like myself, can you quickly explain what those are?
Sure, so non-coding regions, they don't directly code for proteins like the coding regions do, but they control how a gene is expressed, kind of like a volume knob.
Ah, I see. So it's not about changing the song itself, but how loudly it's played.
Exactly.
So by swapping out these non-coding regions, they were essentially changing the volume control for the mouse tert gene.
Precisely. And they specifically targeted the promoter region, which is basically like a landing pad for the proteins that activate a gene.
Clever. So they essentially reprogrammed the mouse cells to use the human instructions for telomerase.
Yes. They tricked the mouse cells into speaking the human language, so to speak.
And what was the outcome of this genetic switcheroo? Did it actually change how telomerase was working in these hut mice?
It did. And it worked beautifully. They found that in the adult hut mice, the telomerase activity was mainly in the testes, the ovaries, and the thymus.
Just like in humans.
Exactly. And other tissues had very low or undetectable activity, totally different from regular mice.
So they basically engineered mice with a human-like telomerase on-off switch. That's pretty amazing.
Yeah.
But the big question is, what effect did this have on their telomere length?
That's where things get really interesting, and we'll talk about that after a quick break. So to get those human-like telomere lengths, it took a few generations of breeding.
Ah, so this wasn't an instant transformation.
No, it was a process. First, they crossed mice with that humanized tert gene with mice that were completely lacking the regular mouse version of the gene.
Okay, so they were essentially combining those modified genes with the complete absence of the original mouse version. What was the next step?
Then they took the offspring from those mice and bred them together. And with each generation, the average telomere length went down and down until it finally settled in that range that's similar to humans, around 10 to 12 kilobases.
Wow, that's incredible. Wild-type mice usually have telomeres around 50 kilobases, so that's a huge difference.
Yeah.
But I'm curious, did having shorter telomeres cause any problems for the mice? Did it affect their health in any way?
That's a really good question, and you might be surprised to hear that for the most part, those hut mice, even with those shorter telomeres, were actually pretty healthy.
That's good to hear. I was kind of imagining all sorts of problems with their cells not being able to divide properly. But I'm guessing there's more to the story, right? It can't be all sunshine and roses.
You're right, there are some nuances here. So while the mice seemed healthy on the surface, there were some hints that those shorter telomeres might have some functional consequences.
Okay, that makes sense. So what kind of functional consequences are we talking about? Can you give us an example?
Sure. So they compared those hut mice to mice that didn't have any functional TERT gene at all.
Okay, so those mice had no telomerase activity whatsoever. What did they find?
Well, in the mice without any telomerase, they saw a much higher rate of germ cell loss in the testes. But in those hut mice, fewer of those cells were lost, which suggests that even a small amount of that human-like telomerase activity was enough to offer some protection.
So the humanized telomerase was making a positive difference, even if it wasn't totally preventing telomere shortening. Were there any downsides to having these shorter telomeres, any negative consequences that they observed?
Yeah, so to explore this further, they exposed the mice to a chemical called DSS. It basically causes damage in the colon, kind of like what happens in ulcerative colitis.
Oh, wow. So they were basically putting those mice through a tough challenge, testing their ability to repair tissue damage. What did they see?
Well, as you'd expect, that DSS caused inflammation and damage in their colons. But the point was to see how well they could recover from that damage, how well their colons could regenerate.
Got it. So did the hut mice with those shorter telomeres regenerate as well as the regular mice?
Well, the regular mice with their longer telomeres bounced back pretty well. They had lots of new cells being made to repair the damage.
Yeah, makes sense.
But the hut mice, those with the human-like shorter telomeres, they didn't have quite the same level of regeneration. I mean, their colons did repair, but it just seemed like the process was less efficient. They produced fewer new cells.
So it seems like their ability to recover from that injury was somewhat hampered by those shorter telomeres. Could this be because their cells were closer to that point where they can no longer divide?
Exactly. That's what we call replicative senescence, and it's a key feature of aging. Basically, cells with shorter telomeres have less room to divide before they hit that limit.
That makes sense. So it seems like there's a bit of a trade-off here. The human-like telomeres in the hut mice provided some protection, but they also came with some downsides. To get a more detailed picture of what was going on at the molecular level, the researchers looked at gene expression patterns in the mice.
Yeah, they did, and this helped them understand exactly how that humanized TERT gene was behaving in those mice. Remember how we talked about how it was mostly active in those specific tissues, just like in humans?
Right, the testes, ovaries, and thymus.
Exactly. Well, they confirmed this by looking at TERT expression levels. They found TERT mRNA in the intestines of regular mice, but not in the hut mice.
So it looks like that human promoter they put in was really doing its job, keeping that telomerase activity under control. But if those cells weren't expressing TERT, does that mean they weren't dividing at all?
Not necessarily. They also looked at genes that are involved in cell proliferation and senescence, and they found markers of cell proliferation in the intestines of all the mice.
Interesting. So even with those shorter telomeres, the hut mice were still able to make new cells in their intestines. But I imagine there must have been some differences at the molecular level, right? Like, were those cells showing any signs of stress or aging?
You're right. When they looked at genes that are associated with senescence, things like P16 and FORA and IL-6, they found something really interesting. These genes were upregulated in those mice that were completely lacking telomerase.
Okay, so that makes sense. Those mice had no way to replenish their telomeres at all. But did they see the same thing in the hut mice?
That's the key point they did. They found those same senescence-associated genes were also upregulated in those hut mice.
So even though those hut mice weren't showing any outward signs of aging yet, the cells in their intestines were experiencing more stress and potentially entering senescence earlier than the regular mice with longer telomeres. Wow, it's like we're getting a glimpse of those very early subtle changes that might be happening in our own bodies as we age. And these hut mice are giving us a way to study those early changes in a more controlled setting. But before we wrap up, I want to go back to those DSS experiments. I'm still thinking about those DSS experiments and how the hut mice, with their shorter, more human-like telomeres, didn't seem to recover from that challenge as efficiently. What's the significance of that finding? Do you think it has any implications for how we understand tissue regeneration and aging in humans?
Well, it definitely suggests that telomere length is a major player in the body's capacity for repair and regeneration, especially when it's dealing with damage or stress. The hut mice did recover eventually, but their slower regeneration could have implications down the line.
It makes you wonder, you know, if someone has shorter telomeres, does that mean they might be more prone to chronic inflammation or slower to heal from injuries? It's a fascinating thought.
It really is. And while we can't make any direct connections to humans based on just this mouse study, it definitely highlights just how important it is to understand how telomere length influences tissue regeneration. And this becomes even more important as we get older because telomeres naturally shorten over time.
So it seems like these hut mice could be a really powerful tool for researchers who are studying age-related diseases and conditions, especially those where tissue regeneration is impaired.
Absolutely. Think about conditions like heart disease or neurodegenerative disorders. Having a model that reflects human telomere biology more accurately could lead to more relevant and impactful research findings.
This deep dive has really been an eye-opener. We've explored the complexities of telomeres and their influence on aging. We've talked about the genetic engineering behind these hut mice, the impact of shorter telomeres on how their cells function, and how well they can regenerate tissue. What do you think is the most groundbreaking aspect of this research?
Well, I think the creation of this mouse model is a huge leap forward for aging research. Having this model that closely mimics human telomere biology opens up a whole new world of possibilities for studying age-related diseases, for testing new therapies, and just for better understanding the link between telomeres and how we age. It's exciting to think about what other discoveries await us as we keep exploring the mysteries of telomeres and their role in human health.
That's a great point. This has been a truly thought-provoking journey into cutting-edge science. And for all of our listeners out there, even though we can't just tweak our telomeres right now, there are things you can do to support healthy aging, like maintaining a healthy lifestyle and managing stress. Until next time, stay curious, everyone, and keep those brains buzzing.